acknowledge the territory in which we gather as the ancestral homelands of the Beothuk and the island of Newfoundland as the ancestral homeland of the Mi'kmaq and the Beothuk. We would also like to recognize the Inuit of Nunavut and Nunavut and the Innu of Nitsanan and their ancestors as the original peoples of Labrador. We strive for respectful relationship with all the people of this province as we search for collective healing and true reconciliation and honor this beautiful land together. Our speaker today is Dr. Karin Limburg from SUNY College of Environmental Science and Forestry. Karin Limburg is a SUNY Distinguished Professor of Fisheries and Ecosystem Science at the State University of New York College of Environmental Science and Forestry, and a visiting professor at the Department of Aquatic Resources at the Swedish University of Agricultural Sciences. Dr. Limburg received her bachelor's degree from Vassar College and a master's of science degree from the University of Florida, and earned her doctorate at Cornell University under Simon Levin. Dr. Limburg's studies have tended towards the transdisciplinary, incorporating physics or economics into ecological research. While the Hudson River estuary and watershed have served as a home base of study, her research expands globally into the Baltic Sea and other regions around the world. With that, I'll now pass it over to Dr. Limburg.
Thanks a lot, Aina. And I guess I'll go ahead and share my screen. Thanks for inviting me. What a wonderful way to visit Newfoundland without having to fly there. So let me just bring this up, and I am going to see if I can minimize things just a moment. Okay, so one thing I wanted to share with the audience is how I remember Einat. This is how I remember her. This is on a field trip in fisheries class. She took the class from me, and this was in 2010. And she's smiling because she has I think it's a sand shrimp on her, adorning her arm there. And she thought it was so cool. So I thought you guys would like to see that. Hopefully that's not embarrassing. It's a nice picture, I think. And let's see, so then onward to what I'm going to talk about today. So some of you may know something about fishes, and others of you may not know much at all. So I'll try to, hopefully I can engage both types of audiences. Given this talk, or forms of this talk in a few different ways and venues, but you'll see here at the bottom of my screen, you can see a little drawing I made. It looks like a fish maybe sitting on a carpet, but it's supposed to be a fish sitting on top of an otolith. And the way I like to introduce a talk like this is to say, imagine if a fish swam up to you and sat down and pulled out a book and started reading to you about the story of its life. That is kind of how I feel what I've learned is about fishes through their hard parts, particularly their ear stones or otoliths, really has been very revealing. I've learned a lot in this very sort of strange corner of biology. There's an emerging field called sclerochronology. Some of you may be familiar with it, but it's making use of the growth and chemical signals in hard parts that have some kind of recording of the environment. And there's in fact a group called, an informal group called the sclerochronologists who have conferences every couple of years or so. And in fact, they're going to have one this year. But among those are these little objects up here in the upper right next to the US penny. Those are all otoliths. And so otoliths have been something I've been studying and working on since my PhD days. It's not the only thing I work on, but it certainly has taken up a lot of my interest. And they're part of the hearing and balance system in the modern teleost fishes. So this is a semicircular canal right here, and there are little chambers below the semicircular canals that contain these little ear stones, and they sit inside these fluid-filled chambers that are also impregnated with nerve cells on the outside, and so as the otoliths move in, as the fish moves, the otoliths rock back and forth in these chambers and bang on the walls and transmit information to the brain, and they're involved both in balance and in hearing. Although I will say that the complete story on how they work for hearing I don't think it has been really solved yet. But there are 3 pairs of them for the modern fishes. But also, I'm going to see if I can just close this window here. Hide the floating window. Yes, I want to hide it. So they come in a lot of different sizes and shapes. They have to conform to the different constraints within the skulls of different fishes. So there are many different types. And you can see these are all from adult fishes, and you can see that there's quite a variety. This is a cod, an otolith from an old cod. These are from alewife. This is from a freshwater drum. These are carp, and so on. Just lots of different shapes and sizes. But for fisheries scientists, they have this advantage that they are really built-in chronometers in fish heads, and they accrete time at 2 different scales. So here's a larval fish, an American shad, and it is growing. The otoliths are actually right here. You can see that in this cross-polarized light image. And they grow daily, on a daily basis. And in fact, when they're growing really well and fast, they even lay down subdaily rings. But you can see that you can actually count these and get an idea of how old a fish is to the day for young fish, which really revolutionized early life history of fish ecology. Then salmon. This salmon is showing here. The otoliths of the salmon are sitting in here, under where the brain would have been. So this is the only really gross picture I think I have in this talk. But the annual banding here can be seen from a sequence of opaque and translucent zones, and that has really lent itself to aging fish. And so, in fact, age determination is really what is mostly done with otoliths around the world. So there are labs all over the world. There are probably some even right there on Newfoundland that have people who are paid full-time to age fish. And the age determinations that they make are fed into models that help with assessing the status of stocks of fishes, which allows people then to set quotas and try to manage fisheries as best they can. So here's a cod otolith. And here what I've done is have a diagram showing that we can make a cut through them, which is called, in this case, in this orientation, it's a transverse section. And what we see on the lower right is an image of cutting through one of these otoliths, and you see these nice translucent and opaque zones. But what's really amazing is that you've got this record from basically when the fish is born and early childhood all the way through life, sort of embedded in this otolith right to the day it died. And so there's really chemical information that's stored inside here, and people like me have been trying to understand and interpret and then apply that kind of information to a variety of different topics. And I'll give you a few topics that I've worked on. But really, as far as these chemical calendar clocks go, sometimes there's a bit of guesswork in it. The time doesn't always appear quite the way we think it might. So some of it is a little bit, sometimes things can be a little bit tricky too. So I'd like to show this slide as well. I'm not going to go into great detail on the kinds of chemical methods that we do in great detail. I just want to say that when we have something like a 2-dimensional object that we're scanning across, we can do some 2-dimensional mapping of the chemistry. So this image in the center was a strontium map that I did at the synchrotron at Cornell University. So synchrotrons are these big, things that big facilities that have do experiments with various elemental particles and nuclear particles and X-rays, and you can do some very interesting, many different kinds of interesting chemistry with them. But scanning fluorescence microscopy is one of them. We also can use a laser and laze across a transect like this and get out line transects. That would be done, in our case, we do it with an instrument listed up here. Laser ablation unit is a high, very fine scale laser, and it's hooked up to another instrument that's called an inductively coupled plasma mass spectrometer. And even with those nowadays, people are starting to do more and more imaging too. So this is actually done with that mass spectrometer. So they're very different types of physics, but they can produce visual estimates of concentrations, and that's really helpful for people, I think, because we're very visually oriented. And so I like to think that we can actually ask questions or have the fish tell us things like, where did I go? And what did I see? And maybe even, was I happy? Maybe. So some examples then of some of this hard part chemistry that I've been working on the last few years, I thought it would be interesting to share with you today. One is I've been involved in some studies with other teams looking at fishes that undertake very large scale migrations and in large river systems and encounter hydropower. The next thing will be ocean deoxygenation related to that, mercury uptake, and then maybe a hint of we may be able to see some signal of ocean acidification. Jury's kind of still out on that. So for those of you who think about rivers and fishes, up where you are, there should be migratory fishes in your streams probably in May and June, something like that. It turns out that fishes need connectivity, particularly the fishes that move between the ocean and inland waters. They need to get up very often to headwater places and rivers. And when there's hydro development, it doesn't always work too well together. So it really is a big threat to connectivity, and it's a very active area of debate. Unfortunately, I think big industry tends to win out, but not always. So these are a couple of examples that I'm going to be giving. One is in the Mekong River, and one is in the Amazon. So this gets to the question of where did I go? The fish that need to migrate, and in fact, I think that many fishes need to migrate, even in of course, in the ocean, but even in small streams. So here's an example. This is a fish called a Goliath catfish. And these fishes are really, really marathon migrators. They spawn up in the headwaters of the Amazon, right along where all these hydro dams are either operational or planned. And then they, when they reproduce, the larvae actually make a very rapid migration by riding the whitewaters down to the estuaries and spend years down in the estuary and then gradually work their way back up. Another very large catfish is one that's called a Pangasius Kremps catfish in a family called the Pangasids in the Mekong River. And the situation in the Mekong River is that there, as you can see, there are many dams that are either operational in the white or red being planned. So it's a very built out system. Excuse me. And the rationale has been that fish don't don't move. Not very much has been known about fish movement in the Mekong River. So as a consequence, the developers say, well, it doesn't matter if we put a dam in here because the fish aren't moving around. But the people I've been working with are proving that wrong. So we can use biomarkers to trace migration between headwater and spawning areas. And we did it, with two different kinds of markers. One is looking at isotopic ratios of strontium. And that's based on looking at the relative age of radioactive decay of rubidium to strontium. And so that's a marker that is one that's widely used in geochemistry. And then second, the bulk trace elements, just bulk strontium and selenium, turn out to be markers we could use. So here we have a situation where we actually were collaborating. When I say we, Anat might remember Professor Don Stewart, who has recently retired as professor of fisheries and ichthyology at our department, and he had a student who I ended up collaborating with, him and his student Ted, but they had actually been discussing with another group down in Brazil and in Peru and France, who had been using the tool of otolith strontium to track migration. And this is kind of a funky graph, because what you're seeing here is a graph set on its side. So this is the otolith image. The little white bars are the annual growth bands. It's where the years are counted. And then this red line is the graph of the strontium isotope ratio put into on its side, so that you would read it from the bottom of the graph to the top of the graph. And what we have here is higher values are on the left on this graph axis, and then they drop down. And what happens is that the fish are born in these headwater areas with pretty high strontium isotope ratios. They drop very rapidly down to nearly seawater levels, and then they gradually move back up again over the last few years. So they're coming back up to about the same level, and so they're actually homing up to. They're going down to the estuary, spending a number of years, and then they migrate back up to spawn. So that's homing. We added on, so Ted and I added on another layer of evidence on this by conducting our own studies at the Cornell synchrotron, mapping out strontium and selenium, and we ratio it to calcium as the major element in the otolith. And again, you can see some of the annual growth bands, not maybe quite as clearly, but you can see these different episodes, and blue is low concentrations, red is high concentrations on this scale. And the big dark lines are where the laser for the isotope, the strontium isotope, was such a strong laser that it just blasted right through. So you've got those big lines there where the material was completely removed. But what we can see is that in the selenium map, for example, it's a little bit obscured, but there's elevated selenium in the core region and then also at the outer part. So that also helps to confirm that these fish were migrating back and forth. And then also the strontium, it's a little bit obscured here, but it was low in the beginning of life and then immediately switches over to being intermediate. So these yellows and reds indicate that this is time spent in the estuary. And then the last couple of years or so, they move up. So that's homing behavior to this upper Madeira River. And in contrast to that, there were fish that were collected that were behind the new dam, and they show no evidence of moving into the estuary at all. So we can then conclude here that otoliths can tell us something about how fishes are going to change their lives as we build dams in large rivers. Same thing with Southeast Asia and the Mekong River and other rivers, the Irrawaddy. My colleagues are also studying that. So this is the Krebs catfish, which, as I said, is a pretty important species. And they display also very, very variable life histories, as it turns out. But this the small blue dots are from on the right hand side graphs in strontium show freshwater signatures, and these fish are migrate 700 kilometers up to the first big impassable waterfalls, which I showed a few slides ago, called Kona Falls, and that's where they spawn. And of course, dams being built right around there right now. But they basically spawn there and then move down. And most of them that we've studied have undertaken complex migrations. But you can see here is a fish that was actually caught back up in freshwater, the top graph, and had spawned in freshwater, moved down river and into the estuary and even into the ocean, and then back up and into fresh water. The selenium is a little bit more complicated, and we think the selenium might have something to do with the presence of shrimp farms in the area, because shrimp farms in the lower Mekong are very dense. Our only place of finding selenium dissolved in water was around the delta in one particular place that's loaded with shrimp farms. And selenium is used as part of a water treatment in some of the shrimp farms. So it's a possibility, but it's just an intriguing thought, really, at this point. But we found that was some initial work that I did before we got a grant from the Australian synchrotron, and I traveled over to work with my colleagues over there in just the summer before COVID hit. These synchrotrons are just enormous facilities. I was working with this guy down in the lower right. His name is An Bu, and he just finished his PhD studying the migration of fishes in the determining what fishes migrate and how they migrate in the Mekong. So it's really groundbreaking work. His advisor here is Lee Baumgartner, and our deceased colleague here, Julia Howitt. But we had and Jason Thiem here. We had a great time spending a few days at the synchrotron, and it's a very powerful synchrotron. So we were able to get great resolution on our analyses. So I'm going to just show a couple of the images that we were able to take, and of course the images are striking, and of course there's data. They actually represent data. So in this scheme, in this color scheme, this sort of whitish yellow is the high, and the blues and reds are kind of lower. But this one is from a high value eel, Anguilla marmorata, the marbled eel, and it's this one was just a phenomenal thing. I called it the galactic eel otolith, just because it's such a cool looking thing. It's elevated in strontium in the core because of being born at sea. These eels spawn on or near seamounts, and probably with high salinity levels. And then as they move into the Mekong River, they apparently make migrations. Either they might make migrations or the river flows are different. But at any rate, we can see some annual seasonality there. This one was another one I thought was really amazing. I called this one the Eye of Sauron. This indicates that this fish did not spend much time in freshwater, but did move into the estuary. But it's a fish, it's another important catfish, but it just happens to be one of the ugliest fish I've ever seen in my life. Luckily, I didn't get to see them live. I just got to play with the otoliths. But every fish we looked at indicated that they had movement in their histories. So these are the top are catfishes. This is another catfish here. this one is another catfish as well, I think, but this is a herring species here. It's a shad. We can see that there's definitely connectivity involved in the lives of many, many species in the Mekong River. So again, hydropower is a threat. There's also some novel, there's maybe novel tracers also that we don't really understand at this point. We were able to visualize iron, which is very difficult to measure with many techniques, bromine and lead. Iron and lead and bromine may have something to do with the redox of the water. So we don't really know, but this is something that's really intriguing and I'd love to follow up on. Okay, so next then is the ocean deoxygenation and its effects. So basically, this is a big topic I've been involved with for a while. Just as an aside, the UN just approved a treaty on biodiversity conservation of the oceans and recognized that oxygen loss is an issue, along with warming temperatures and acidification. So it's getting more attention now. So warming lowers the oxygen solubility. It stratifies. You're probably seeing increased occurrences of this up where you are. There's less ventilation and so on. And for fishes, this leads to some compression of the habitat space available. And when they're more crowded together, you would get density dependent effects. Maybe there's not enough food to consume, for example, there may be starvation. And of course, in extreme cases, there would be death. I do know that you have a chronically hypoxic area near you in the Gulf of St. Lawrence. It's not so far away from you. But it's a growing problem worldwide. And this tale is about cod, the Atlantic cod, which you guys should know quite well up there. And as you know, in the Northwest Atlantic, it's not doing all that well. I'd love to discuss that with some of you afterwards, but it's doing very poorly in the place where I study it, in the Baltic Sea, because the Baltic not only has warming, but it also has hypoxia. So the Baltic Sea, and this is just showing you the kind of what's called the Baltic proper, here, the central Baltic part. It's connected to the North Sea, which would be over on the left-hand side, coming in through the Danish Straits and through all these little seas down here. But the black marks here indicate the surface area at the bottom that's anoxic or even euxenic, and then the gray is hypoxia. And this has been increasing over time. So this map was created in 2019, but it's sort of similar now. And where you've got these big anoxic areas, these tend to be deep zones, and there's very complicated circulation in the Baltic Sea. And just for scale, I'm not showing you the entire Baltic because there's a big bay to the north called the Bothnian Bay. But the entire surface area is about 1.7 times the surface area of all the Great Lakes, the Laurentian North American Great Lakes combined. So it's a very big place. These ellipses that I put here are places that have been, until recently, they were spawning and nursery zones for cod. And so those on this different projection here show you that. So in the 1980s, cod were doing quite well. There were these areas like the Gotland Deep here was a huge spawning area. Another, the Gedans Deep, the Bornholm Deep, and then spawning. And the waters that circulated around the Baltic would transport fish throughout parts of the Baltic Sea. And now from about 2010 onward, we're seeing just a great reduction in spawning here, although we now have some potential evidence of possibly another population up to the north, but that's again a developing story. But as far as we know, most of the compression of the spawning volume has been pretty severe in much of this large inland sea. That's had effects. This is a bit of an exaggeration, but most people show this kind of picture anyway. There were some extremely good year classes of Baltic Sea cod in the 1980s, and they led to some very well-developed population age structure. And this is a fisheries scientist from Estonia hugging a cod, and they were quite large. Today, they're tiny. They have shrunken drastically. So this is really what they look like now. I've dissected some myself. I know that this is true. And the occurrence of very skinny ones, which the Swedes call necktie cod, because that's what they kind of look like, has increased too. That's not every single cod, but there are a lot of cod that are not doing well. So it turns out that fish otoliths, again, can tell us something. And it tells us about fish exposure to hypoxia. And that's because of redox dynamics, that the trace element manganese, which is pretty abundant in the Baltic Basin, it's readily taken up as manganese 2 and manganese 3, and that's dissolved under reducing conditions, which include low dissolved oxygen and also higher acidity, which I think is both are going on in the Baltic. So these are modern cod otoliths. These are Stone Age ones. So I've actually tried to use Stone Age otoliths as kind of a baseline, and then this is a clump of manganese from the Baltic. So, so the idea is that in the Baltic Sea and in many places, we have seasonal hypoxia, and manganese can cycle accordingly with it. So when you have water that's well oxygenated, what we call normoxic water, there's a nice cap of sediment, a centimeter or more of oxygenated sediment, and that basically keeps the manganese inside the sediments, and sometimes they get buried as particulate manganese carbonate and stuff. And this sort of cycling happens in the spring and fall and in the winter. But in the summertime, typically, you have hypoxia setting in, and this oxic sediment layer gets very, very diminished, and it can even disappear. And once you have that happen, you've got a way for a sediment water coupling, and you can have diffusion of manganese across that boundary. So we think that's how this works. And so that we can actually then say, for the second thing, what did I see? Do we see the good, healthy Baltic Sea, or do we see the bad and the ugly? And this is an image of the sulfur bacteria, anoxic sulfur bacteria, Vegeotoa here. So it turns out we can track hypoxia exposure by manganese in the otoliths. So here's an example again from the synchrotron, and it's a little bit speckly because of the low concentrations of manganese, but basically taking averages across the site, what I did was I took a transect with the GIS program across these maps and averaged out the seasonal values of manganese in ratio to calcium and strontium in ratio to calcium. What we see is an elevation in the summertime and then a decline in manganese. And at the same time as that, we see in the young of year fish, we see low strontium and then an elevation in the fall as it moves out to sea. And this is interesting because what we see this kind of pattern in strontium right back to the Stone Age, and it appears to be use of coastal nursery habitats. And so we can see that here in a fish that's it has gone through 2 seasons as well. Well, here's its nursery zone area, and then its hypoxic exposure. So first good water, and then a kind of a hypoxic summer, a second winter, and then pretty bad hypoxic summer. So it looks like this is a pretty good proxy for hypoxia. But the other thing we've learned about a number of different elements that incorporate into otoliths is that some of them are very clearly have something to do with the environment the fish are in. Some are physiologically regulated, and some are kind of in between. And manganese is somewhat in between. So it seems to be growth sensitive. There's still a lot of work that would need to be done to understand exactly why. But it does decline with growth rate. And when I examine fish from normoxic water, from healthy fish, like in Iceland, I can still see this. I can see the presence of manganese is much, much lower than I would see in hypoxia, but it still is relatively high when they're young, and then declines as their growth rate declines as they get big. So aside from manganese, as I say, there are other elements that are under physiological control. And one that got my attention is the trace element magnesium. It's a little confusing to talk about manganese and magnesium, but we'll just try to keep them separate. But it also substitutes for calcium in otolith aragonite. And unlike, in most oceanographers think of magnesium as tightly coupled to salinity, which it is in water, but it doesn't seem to be in fish. It seems to be more linked to metabolic activity, and it shows patterns that are somewhat different from manganese. And so as I started to look into this some more, I stumbled across a very interesting paper that was floating around on the internet and from the Clinical Kidney Journal. And it's a neat paper and describes magnesium dynamics. And one of the things that it talks about is the fact that magnesium, even though it's got a fairly similar radius to calcium, it's doubly hydrated as opposed to singly hydrated calcium. And so because of that, it has to be able to go through ion channels, it has to shed 2 layers of water, not just 1 layer like calcium does. And as they say here, it has to get rid of 2 layers, which is highly energy consuming. And then the other thing I was reading about was I was reading a set of papers by some people from France and Italy and Germany describing sort of the proximal chemistry of the environment that the otoliths are formed in. And they form in these little sacs that I mentioned early on in my talk. And these sacs have ion channels on the top side by the nerve and on the bottom side as well. And it turns out that these ion channels here are associated with tons and tons of mitochondria. And so I thought, aha, this is like a powerhouse. So that's where they are. And so the magnesium is actually located near, when they look at the chemistry of the internal fluids, it's near these powerhouses. And so we came up with a hypothesis that, and we know also that magnesium binds to ATP to activate it, and ATP is intimately involved with the enzyme star maker, which is what biomineralizes the calcium carbonate into aragonite in the otolith. And so we hypothesized that the magnesium ebbs and flows with the seasons of growth. And so it seems to fit well with a metabolic theory of magnesium. And a good example of that is looking at a flounder, Platyctes flessus, which is actually, I call it the Baltic flounder, but it's really a pan-European flounder. And when we do the due diligence, if you will, and what I did for some of my flounder samples is I thinned down the otolith enough that I could count daily increments. And when I did that, I could see that the pattern here of magnesium to calcium, which is very clear in this otolith, the peaks fall exactly on top of the fast-growing bands, the bands that were laid down in the summer, and the valleys fall right down smack on the places where the otolith basically stops growing in the winter. So it's a really good chemical calendar, at least in some fish taxa. And we also can say that for healthy cod, we see clear patterns, and with fish in very poor condition, we see very poor patterns. And I had a PhD student in Sweden who's made a study of this for her PhD to see whether or not we could use the chemistry of otoliths to help us age the fish because they're growing so badly in the Baltic that they don't lay down clear increments in many places. And so one of the things that I found as I was going through this, I was working with another colleague over in Sweden, Michaela Cassini, and we were studying cod in hypoxia. And to my sort of surprise, when I started analyzing the cod, we were interested in looking at their condition. And I found that the condition, we split them basically down the middle into low condition and high condition fish based on a fatness index called the Fulton condition factor. And when I did that, I was quite surprised to see that the groups fell out this way, that there's more magnesium to calcium in fish with high condition and kind of a declining level of magnesium as the fish get older and older. And this was striking to me because really these fish are from surveys, fisheries surveys, and what happens is they get caught in these experimental trawls and they get whacked and killed, and then they're measured, their lengths and weights are measured, and that's what they calculate the condition factor on. And that condition factor is calculated exactly once for those fish, but yet their values sort of persist through life. So it sort of looks like this is something that this low condition appears to be something that may have long-term carryover from previous stresses. So magnesium seems to reflect growth and metabolic activity, and it might tell us something about the state of the animal through its life. And it may also be a way to correct this hypoxia proxy for growth effects. So very, very simply, I suggested that we may be able to use manganese over magnesium as our somewhat corrected proxy. I don't think it's perfect, but it does help. When we did that, and then I plotted that against, and I made a plot of, I made a plot of that in these red dots from the data I had and overlaid it on top of a trace of the hypoxic volume that's calculated for the Baltic Sea over time. And what I ended up doing was lagging it so that the low points here would match. And when I did that, I was shocked at the relatively close correlation between these because these are just a small sample of fish, really, but yet their patterns really seem to match pretty well against this other completely independent index of hypoxia. We can also, in fisheries, back calculate the lengths of fish at different ages. And doing that, when I separated them into sort of quartiles of hypoxia exposure in any given year of life, I also saw this great separation. So there's not much separation when they're young 0-year-old fish, but as they get older and older, the separation kind of increases. So the least exposed have the highest growth. The most exposed fish have the lowest growth. And the difference between the two is a 39% decline in growth, in length. And that translates into a 64% decline in weight. And since what fisheries people care about is weight, because that's the fish, that's the fillets that we want to eat, this has huge economic implications. So then just kind of wrapping things up here a little bit, the last part here is talking about a current project that I'm the lead on, as it turns out, called Project Breathless, and we have a website that we have several people contributing to, and it's a pretty nice site. So if you're interested, this is the link to it down here. It's about hypoxia, and our teammates decided to name it Project Breathless. And what it entails several coupled components. One is empirical studies, like I've just shown you, of body parts. Another is a set of size-structured food web models, which is ongoing in the Baltic Sea. We are, as a group, trying to also think about the ecosystem services that are at risk from hypoxia. And in fact, we've been collaborating a little bit with Rashid Sumaila, who's a professor at UBC, who actually just won an extremely prestigious award called the Tyler Prize in Environment with his colleague Daniel Pauly, who you may know, he's also at UBC. And then communicating with different audiences, which has been really fun. I'd love to talk to you about that too. So these are a couple of the collaborators on the project. Roxanne Razavi is an assistant professor in my department I collaborate with, and we co-advise Hadis Marali, who is in the lower picture. And Roxanne is an ecotoxicologist who specializes in mercury dynamics. And mercury turns out to be enhanced in its bioavailability by hypoxia or anoxia. So the microbial communities in anoxia can methylate mercury. And once that's methylated, it's toxic to organisms and not good for humans. And as it once it's methylated, it can also persist in hypoxic conditions. And so there's a mechanism for then being available to a food web. So we've been interested in this coupling too. And the way we do that is by bringing in another chronometric structure. So we also look at islands. And I think those of you who are fisheries interested people in the audience should be given a high, heads up that pretty soon I think there's going to be lots and lots of studies of islands. So here's a pair of otoliths and a pair of islands out of yellow perch. And the islands are interesting because they are a completely different material. So otoliths are made mostly of calcium carbonate precipitated on a little, meshwork of protein. These are 100% pure protein. So they have affinities for different trace elements. And one of the elements that they're really good at sponging up is mercury, in fact, methyl mercury. And there's been some great work done up at the Canadian Light Source working out a lot of this exposure. So Hadis gave a talk recently at a toxicology conference about her first chapter, which is developing a methodology to parse out a lifetime exposure of mercury. And then the paper we're working on together now is how to link that up, how to link up hypoxia to mercury exposure. But I also want to give a little shout out that our paper just got published. Our first paper got published, and we made the front cover of ES&T letters, which was pretty cool. So the study systems there, again, the Baltic Sea, but also two sites in North America, Lake Erie and the St. Lawrence River. And the star of this story is the round goby. And they are invasive. They are primarily found in fresh waters, but they can handle a fair amount of salinity too. And they're very invasive. And they are highly adaptable to many conditions. So they've taken hold in the Great Lakes, in parts of Europe, and they might be heading towards you, who knows, at some point. But what Hadis did is she took otoliths. We know that eye lenses and otoliths both grow proportionately to body size. And so what Hadis did with these round goby otoliths is she marked off where the annulus formations were, and then assign proportionality to the islands, the islands radii too. And so she could then quantify on an annual basis the concentrations, the mean annual concentrations of mercury. And when she did that for these different systems, what she saw was increases over time, bioaccumulation, I guess, over time of mercury concentrations in Lake Erie and the Baltic Sea. But she was a little concerned because she saw a decline in the St. Lawrence River. And the predictions then showed differences with muscle tissue. So on the left, we're seeing whole muscle tissue, which is the kind of the conventional way of measuring mercury exposure. It increased in all three systems, but here we can see that the St. Lawrence Islands mercury declined. But then Roxanne remembered that there was a recent paper published by some of our colleagues that suggested that maybe this has to do with diet changes. And in fact, there's a paper by people actually also from ESF who found that smaller fish had higher on the food web, a higher signature. They were eating organisms that were higher trophically than the older fish. And the older fish started specializing on zebra mussels, which are also an invasive from the same area that the ground gobies come from. And so they were consuming that. And we know that mercury and nitrogen isotopes correlate very well together. So it looks like that's probably what we're seeing. And so this study helps explain our finding of lower mercury in ground goby lenses. And so we have a way of putting a timestamp now on mercury exposure through time, and we're applying this in other systems too. So the last little tidbit here is this question about whether or not we might also be able to see an ocean acidification signal. I only have one slide on this, basically just to say that I started looking at the trace element boron because a colleague had suggested it might be a good signature of declining pH. Boron in carbonates is in the form of borate, and borate correlates positively with pH. And here you can see from a fish I analyzed from the year 2000 that there's some pretty strong patterning in it. I happened then to continue my hypoxia studies and followed also up on the boron in 2019 and saw that this is a dramatic change. And I double checked the analyses, ran the samples again, and they all checked out. So something is really different. And I'm not saying, I don't think I can absolutely say that boron has a lot to do with the pH, but we do know that the boron to calcium ratios are declining in the Baltic right now. And so we don't know yet if it's really and truly a early warning system about ocean acidification there, but it's got something's going on for sure. So I think what I hope I've done is to show you that otoliths are very much like logbooks for individual fish, and that we can learn a lot. We're still learning tons and tons. It's not a closed story by any means, but I think that it's something that many people are going to continue to pursue in our world of understanding the lives of fishes. So with that, I'm going to say thank you very much, and I'm happy to take questions if there are any. Sorry for coughing.
Thank you so much. That was very interesting. I'm going to open it to questions in-house, and then also online. If you have a question, just type it into the chat. If you want to come here, use the mic.
Oh, great. Yeah, it'd be nice to see people.
Yeah.
Okay. Hello, thank you so much for that. Oh, wait, which one? Oh, hey, can you hear me now?
Oh, absolutely. Yep.
All right. Yeah, thank you so much for that. I always find a way to work this in with every speaker. It seems like everyone's tied to Florida somehow, but I worked at UF from 2016 to 2020, so I was over there by Deb Murray and all them for quite a while.
So but in zoology?
Fisheries.
Oh, okay. Oh yeah, over in IFAS. Yeah.
Yeah, exactly. Mm-hmm. I shared a lab with somebody who was working on using non-lethal aging techniques. So they were taking largemouth bass and cutting off all the all the hard parts on the fins and stuff like that. And I was just wondering if you had any experience with that in terms of aging, what your outlook on using those non-invasive techniques or non-lethal techniques, what your outlook on using that for aging would be. And then also in terms of biomarkers, can you use those?
Yeah, that's a couple of great questions. And there's a wonderful review article. I don't know if you've seen it. It was published in, I think, 2017 or something like that. In Limnology and Oceanography Methods, LNO Methods, by a guy named Orion Sadzik and his advisor and other people. And it's a real nice review of all of that. There's a lot of interest, obviously, in non-lethal sampling. I mean, the big downside of doing what I do is that the fish have to be dead. So the answer is sometimes. Sometimes you can do it. So probably with the bass, they can. I'd say those kinds of techniques probably work better with short-lived fishes rather than long-lived fishes, for one thing. And that's something that's been well recognized for quite some time with scales, for example, scale analysis. That scales just if they do grow, they're growing so slowly for longer lived fishes that you can't resolve the growth rings at all. So that's obviously a big problem. And so I have colleagues who are trying now to see if they can do non-lethal sampling for aging eels in Europe, because eels over there are critically endangered. So you don't really want to go killing lots and lots of them. But it's not clear that will work. But there are techniques, to use those other parts. Sturgeon are typically aged by taking a chunk out of their spine. I'm not sure that's necessarily great for the fish either. That probably does some damage as well. But I don't know if there are ways of providing something to stabilize those parts. I mean, even cutting the spiny rays off of bass. I mean, I don't know what the damage is there, but at least I guess you're not killing them anyway. So that's probably a better alternative. But there is, as I said, there's a great review article if you want to pursue that.
That's great. Thank you.
You're welcome.
Another question coming up.
Incidentally, how many of you are in fisheries? Maybe you can maybe you can tell me, you at the microphone can tell me if people are raising their hands. Anybody out there in fisheries?
Hi, Piotr Trela here. You mentioned, the first part about strontium having different levels in, close to the source of the river, estuary, or open ocean. What's the reason? And asking it's because I guess aquatic ecologists are in more difficult situation than land ecologists, because on land, if you have, for instance, stable isotope signature, it stays there. So if you were in one place, you will have one. So in other words, the boundaries between values are much more highly resolved. In a river, if you put some strontium, let's say source of strontium, it will flow with it. So you can't really pinpoint where exactly how much time you spend, let's say, in the first 100 kilometers of river, how much time later. But what's the overall cause for the difference in strontium between rivers, estuaries, and the sea?
Well, in most of in many watersheds, relative to the ocean, they are depleted in strontium. Strontium is an element that substitutes in rock for calcium. So it's what and strontium is taken out of the seawater, I guess, and incorporated into the shells of corals and bivalves and so on. So when those eventually become rock, the strontium is incorporated into the rock. And so that forms I guess, a basis for erosive material that can be, dissolved eventually. And so the variation of strontium in rock and soil determines to some extent the strontium concentrations in fresh water. But fresh waters generally are depleted in strontium relative to the ocean. That's not always the case. For example, here where I live in central New York, there's a lot of ancient marine bedrock, and the strontium is as high here as it would be in the sea. So you really have to know something about the geochemistry of your local surroundings. I'm not sure if that helps, but it's basically, it's mostly related to the watershed dynamics. And there's relatively speaking, the ocean is relatively homogeneous with respect to strontium. Estuaries are quite variable, but typically we can see gradients with lower strontium to calcium ratios in the fresher zones and more in the marine end members. So we use mixing diagrams just like one would with salinity to look at the rate of change of strontium to calcium with salinity.
And another question, if I understood correctly, you mentioned that you were expecting the difference in a magnesium to calcium ratio as a function of salinity, but you didn't see it. But from my experience in oceanography, you're looking for magnesium as calcium as a paleothermometer. So effectively, it changes with the temperature. So with fresh water, I mean, it shouldn't affect if you have things like not like a Baltic Sea, so open ocean, when you have mainly evaporation and precipitation, which does not change the ratio. In the Baltic Sea, I mean, if in a river water the ratio is very different than in the ocean, then it will be confounded. The signal will be confounded, so they will be affected. Should be affected by the source of the water there. And so, but I mean, maybe some of that. I mean, this is quite interesting. I mean, you've shown that there is the poorly conditioned have a lower and lower time with life for the ratio. I mean, if they were in poor conditions because they hang out in the colder water, this may have, for longer period, this may have explained why they were dropped in calcium, in the magnesium relative to calcium.
Right. I don't think we have any demonstrative proof that these fish that the fish that were in that sample came from warmer or colder water. So I think that they mixed. And what really convinced me that there was something going on, I think I actually have a diagram in a different part of my talk that I didn't show, but basically there are times in healthy cod, and I'm speaking really from cod and flounder and that kind of thing, and it's not I can't speak across all the 37,000 species of fish, but for some taxa anyway, we can see that magnesium and manganese cycle together seasonally in healthy fish. And then what I was observing in these cod in the Baltic was at some point when the fish were older and were experiencing great amounts of hypoxia, the magnesium would just sort of disappear. It would just start to sort of just go away, decline a lot. And it was like these two physiological properties were sort of decoupling, I guess. So, it's a difficult thing when you have an element like manganese that has some sort of a physiological regulation as well as the presence in the environment. But what I can tell you is we've seen plenty of fish where we see manganese during a period of moderate hypoxia, and then the Baltic Sea has an interesting history in which in the early 1990s it became very well oxygenated, and in that time when you scan an otolith that occurred during that period, you've got a temporal marker where the manganese just disappears and the magnesium shoots up. So it's very interesting that way. And I don't think it's warmer.
Okay, so just very quick last question.
Mhm.
Do you think, I mean, the composition of otoliths, would it be affected? I mean, where the atoms or isotopes into this go? Do they go from water they drank, or do they come from a food they ate? Because then
Yes.
maybe different path.
Yes, both.
Both.
We think both. So your former countryman, Steve Campana, wrote a very interesting paper in 1999 about otolith chemistry, and the consensus has been that most of these trace elements enter through drinking the water, passing it over the gills, but there has to be some absorption from the gut too, and we know that that's the case. But I think there's really a real shortage of good tracer experiments, and of course, the tracer experiments I'd love to do would be with manganese, but manganese has only one stable isotope, and so it would be kind of it would be difficult, I think, to get the permission to use radioisotopes to do the experiments that one would want to do.
Thank you.
Thank you for your questions.
Thanks. To answer your previous question, I think almost everyone here is working on some aspect of fisheries science, from modeling to contaminants and habitat studies.
Great.
So very well represented here. No, it's a previous. Sorry. One question about islands. Is there any limitation for the size of fish, or is it like otoliths where larvae could be used?
Well, they're pretty darn small, you know, when they're in a larva. I've never tried to dissect them out of a larva. I think some people have. For trace element work, I guess you really are limited sort of by the size of the material you've got. But one thing I didn't mention, and people should take note if they're interested at all at looking at lifetime histories of stable isotopes, is that being pure protein, they have all of your favorite light stable isotopes, and people have demonstrated again and again, looking at trophic shifts but you can plot out the chronology of shifting from being a plantivore, for example, to a piscivore by peeling layers of the islands and shipping that off to a stable isotope facility. So we are looking in our Project Breathless at 3 isotopes in islands: is carbon, nitrogen, and sulfur. Sulfur is another sulfur isotope ratios are sensitive to hypoxia, or to well, I guess you could say hypoxia. So it's when sulfate is reduced to hydrogen sulfide, there's a massive drop in the sulfur stable isotopes. And although we don't see massive shifts, we do see shifts. We do see declines in hypoxic systems. So, and I imagine the reason we don't see massive shifts is probably because fish are consuming different things, different food items, and moving in and out of hypoxia most likely. So, but we are seeing that, and yeah, it's an interesting set of markers. But presumably, you could look at the oxygen isotopes. I'm not sure how easy it would be. I don't think you can use oxygen in organic matter to look at temperature, for example. But you can look at deuterium. Deuterium is a nice isotope for separating allochthonous production from autochthonous production in rivers, for example. And I'm sure there's many other applications. So there's a lot of things you could do with eye lenses, or people will probably be doing sooner or later. So, I mean, as long as if you've killed the fish for its otoliths, you may as well take the eye lenses too, I say, right?
Mm-hmm. Okay, cool, thank you.
Sure.
I think that's all the time we have for today. So thank you again for a great talk, and for everyone that joined us. Next seminar will be Thursday, March 23rd, where we will hear from Dr. Dan Duplisa from DFO about incorporating climate change into fisheries self-management.
Well, give him a big hello from me because we overlapped at Stockholm University. So,
Oh, cool.
yep.
I'll
it's
send
a
you the
small
link.
world.
You can join the talk too.
Good. Well, hi, Dan. All right. I'll see you guys.
Thank you.
Bye-bye. Thanks for inviting me.